So pleasant day, dear doctors, and welcome to our module. This is a supplemental session, which will focus on biochemistry, but it will have a touch of pathology in pediatrics. And we're going to focus on a chapter which recently had several key points that popped out in the exam. And this is under the chapter Intracellular Traffic and Sorting of Proteins. So this is Dr. Toom. Let's begin our session. These are the following learning objectives for today's session. So first, we're going to reintroduce an organelle we've learned in the past, which is the endoplasmic reticulum, as well as the Golgi apparatus. What actually is the role of these two organelles in the sorting of proteins? We will also recognize special signals, which basically involve other cellular organelles like the mitochondria, the nucleus, and the peroxisomes. We will also introduce and explain the role of chaperones as well as ubiquitin, and we will lastly discuss diseases which result from the mutation of genes which encode proteins which are involved in intracellular transport. 90% of the lesson is lifted from Harper's Biochemistry with supplemental key figures and clinical applications lifted from Integrated Biochemistry by Dr. Michael King. Now, these are the organelles which are involved in the transport, the intercellular transport of proteins. So number one, we have the Golgi apparatus. We have the endoplasmic reticulum. We have the mitochondria. And we have the nucleus. So kindly repeat this to yourself right now. What are the organelles? What are the organelles involved in intracellular protein transport? We have the endoplasmic reticulum, we have the nucleus, we have the mitochondria, and we have the Golgi complex or the Golgi apparatus. Now, question: which part of the endoplasmic reticulum does not play a role in protein synthesis? Which part of the endoplasmic reticulum? The endoplasmic reticulum has two parts. It's the rough endoplasmic reticulum, or the rough ER, and the smooth endoplasmic reticulum, or the smooth ER. The smooth ER does not play a role in protein synthesis. It plays a role in lipid metabolism. Okay? It plays a role in fat metabolism, as well as the metabolism of steroids. Those are just some of the functions of the smooth endoplasmic reticulum. Now, I want everyone to remember that from the endoplasmic reticulum, it's going to reach the Golgi apparatus. And the Golgi actually histologically has several parts or layers. We have the cis Golgi and the trans Golgi. The Golgi apparatus will serve as the processing and the packaging center. Processing and the packaging center of the cell. It is from the Golgi apparatus that contents such as protein can reach the cell surface, or they are degraded in the lysosomes. The lysosomes are the digestive organelle. They are the digestive organelle of the cell. Or from the Golgi, these proteins or other substances will be stored in vesicles. So the Golgi is for the processing, the packaging, okay, the sorting of proteins. So if this was a company, an industry. The Golgi apparatus is like the assembly line. It's the assembly and distribution line of a company. Okay. Now, I want everyone to be familiar that the endoplasmic reticulum is here. Okay. From the cytosol, proteins, we're going to zoom into proteins, will be processed in the endoplasmic reticulum. This is an example of co-translational transport. From the endoplasmic reticulum, it reaches the Golgi, and from the Golgi, it will be distributed to the different parts of the cell. From the cytosol, okay, we have what we call post-translational transport, wherein substances like proteins can be brought to the mitochondria, they can be brought to the peroxisomes, or they can be brought either to the nucleus. Okay, so there are several pathways. Which proteins will pass through. Okay, now this is what I want you to be familiar with. Let's go textbook. In the synthesis of specific proteins, okay, there are basically going to be two forms of synthesis. First is the cytosolic. Second is synthesis through the membrane-bound polyribosomes. If it is not membrane-bound or cytosolic, this is what we call free cytosolic or free synthesis. So these are the two major forms by which proteins can be synthesized. Now, I have to mention this because this is found in Harper's. There's a famous doctrine or hypothesis which we call the signal. Okay, we call this the signal hypothesis. Now, what I want you to remember is that there is what we call a signal peptide, and this is known as the N-terminal signal peptide, or NTSP. N-terminal signal peptide. This is found in the polyribosomes. So what happens is proteins that are synthesized on these polyribosomes contain this peptide. And this will cause these peptides to attach to the membranes of the endoplasmic reticulum. And it will help facilitate the transfer of these proteins. So what you have to remember is that proteins which are synthesized in the free polyribosomes, they lack the signal peptide, and therefore they will retain their movement within the cytosol. So please take note of that. Okay. Now, sorting of proteins. So from the polyribosomes, we have what we call the cytosolic and the rough endoplasmic reticulum. These are the types of proteins which will be sorted in the cytosolic branch. These are the mitochondrial proteins, nuclear proteins, peroxisomal proteins, and cytosolic proteins. While proteins that will be sorted in the rough endoplasmic reticulum would be the membrane proteins, okay, the endoplasmic reticulum and the Golgi apparatus proteins, as well as the plasma membrane proteins. The rough ER is also the branch where secretory and lysosomal proteins, okay, secretory as well as lysosomal proteins will be sorted. Okay, so I want everyone to remember that. Now, this is a very important table lifted from your Harper's. It shows you What is the targeting sequence or the compound and the specific organelle that is targeted? I want you to remember only two things here. The endoplasmic reticulum, okay, the entire endoplasmic reticulum, okay, that is targeted by the N-terminal signal peptide. And lastly, this one came out, but this came out in patho, but they presented it as a lysosomal storage disease. They were asking what is, okay, they were asking what is the targeting sequence, which is defective or deficient in lysosomal storage diseases. And they mentioned specifically eye cell disease. This is kind of deep in your textbook, okay? But it's something which I want you to remember, okay? That is mannose-6 phosphate. That came out about 3 exams ago. A mannose 5-phosphate. Okay, so there's 2 things I want you to bring here. Okay, now, what is the role of the endoplasmic reticulum and the Golgi apparatus? So let's focus on the Golgi right now. So the Golgi apparatus plays a role in what we call glycosylation and sorting of proteins. So I want you to remember that these are the 2 important roles of the Golgi apparatus when it comes to protein synthesis. What are those 2 roles again? I want it to come from you, the roles of the Golgi apparatus, because everyone memorizes the packaging. It is glycosylation, okay, and it is sorting of proteins, okay? And please take note that the Golgi apparatus is involved in the processing, particularly of oligosaccharide chains, okay? So this came out in the boards, and they asked about collagen. Collagen is an example of a protein that undergoes significant glycosylation as well as sorting. Collagen is actually comprised of what amino acids? What amino acids is collagen comprised of? Let's see your answers. What are the collagen amino acid sequence? Okay. So what is the amino acid sequence of collagen? Okay, very good. It has glycine. Okay. It has proline. And it has hydroxyproline. Okay. Everyone has to memorize that. Okay. Everyone has to memorize that. So please take note of this. Okay. Now, we mentioned cis and trans earlier. I would like to remind everyone that the Golgi apparatus has these parts, okay? It has the cis, or here's the cis Golgi. This is the side that faces the endoplasmic reticulum. The trans, which is here, Okay, call the TGN. That is the trans-Golgi network. Okay, that is the one that faces the plasma membrane. And of course, we have the medial part, which is the one right in the middle. Okay, so that is the meaning. That is the meaning of cis and trans. So please take note of that. Now, let me show you this. Protein synthesis is here. And protein synthesis leads or yields unfolded proteins. So unfolded proteins need to undergo folding. Unfolded proteins are supposed to be folded. And once proteins are folded, they are now removed so that they can be utilized. Now, question. What type of protein is normal and what type of protein should we have? Should it be the folded or the unfolded form? I want to go very, very basic to check if you understand. What should our body use? Should it be the folded or the unfolded form? It should be the folded form. Unfolded proteins that don't undergo folding, or we have misfolding of proteins, which usually occurs because of mutations. This leads to aggregation. Okay, this leads to aggregation. So there's now protein aggregates. Now, what is responsible for the proper folding of proteins? What is responsible for the proper folding of proteins? So we have what we call the chaperones. Now, can you give me a disease which results from a misfolding of proteins and leads to the accumulation of tau proteins or aggregates of tau protein? What disease is that? There is now going to be an increase of aggregates of tau protein. Okay, this is your classic Alzheimer's disease. Okay, that is classic Alzheimer's disease. Okay, so this is quite important. Now, the chaperones or the chaperonines, okay, in our Harpers, the chaperonines are now considered to be a second major class of chaperones. Okay, we call them chaperonines. Okay. Now, chaperonines have a unique structure. They are shaped like barrels, barrel-like structures. Okay. Now, I want you to remember that chaperonines have subunits. And one of the most important subunits there is what we call the heat shock protein, or let's refer this to as HSP 60. Okay, we will talk about this in a while, but I want you to remember this is how you top the exam. You are digging the major chapters in the book. So I want everyone to be familiar with heat shock protein. Okay. So here are just some illustrations. So here's the HSP 60. If you look at the structure, okay, they actually look like they are barrel shaped. Okay, these are chaperonines. Now here's a table in your textbook. Properties of chaperone proteins. So they are found in bacteria, they are found in humans, and we refer to chaperone proteins as HSP or heat shock proteins. Okay? So please take note of that. That's what I want you to know there. Now, mitochondrial proteins are usually imported. That means they are not synthesized in the mitochondria. If you recall earlier, we mentioned post-translational modification involves transport of proteins from the cytosol going to the mitochondria. Now, most of the proteins which are found in the mitochondria, these are mitochondrial proteins, they are actually encoded by what we call nuclear genes. So encoding is done by nuclear genes, and they are actually synthesized outside of the mitochondria, and they are imported. Of all of the mitochondrial proteins, the one that I'm going to bet my license can pop out in an exam are the subunits of ATP synthase. Now, question. What pathway or metabolic pathway would you encounter ATP synthase? Where would you encounter ATP synthase? Where? Okay, this is found in the electron transport chain. Would you happen to know what the other name of the electron transport chain is? What is the other name of the electron transport chain? The electron transport chain is also known as the respiratory chain. Okay, this occurs in the inner mitochondria. Now, how many complexes does the electron transport chain have? How many complexes does the electron transport chain have? It has 5. Which complex is ATP synthase? Okay, very good. This is complex 5. Okay, that is complex 5. Now, another process in the sorting and transport of proteins is what we call translocation. When we say translocation, this refers to the passage of proteins from one membrane to another. Okay, in this case, it would be the outer and the inner mitochondrial membrane. Okay, so let's just say this is the mitochondria. Inside, okay, you have the cristae, the inner mitochondrial membrane in the inner mitochondrial fold. So from this is the outer mitochondrial membrane. This is the inner mitochondrial membrane. The transport or passage of one matrix protein from one membrane to the other, that is translocation. Okay, that is translocation. So here, this is what we call the TOM, not tomb, but TOM, which means the translocase of the outer membrane, and the TIM, TOM and TIM, translocase of the inner mitochondrial membrane. Okay, and you have here an example of your heat shock proteins. Okay, so I hope everyone understands that proteins, remember, are proteins synthesized inside the mitochondria? Yes or no? Are proteins synthesized inside the mitochondria? What is your answer? Are they synthesized in the mitochondria? Yes or no? The answer is no. Okay? That is why they need to be transported to the mitochondria. And these proteins will pass from the inner to the outer mitochondrial membrane. And that process is known as, fill in the blanks, what we call the passage of a protein, okay, from the inner to the outer mitochondrial membrane. That is called translocation. Now, here are just some examples of the terms we've been mentioning. So here, the translocase of the outer membrane, that's what we call TOM. And what do we call the translocase of the inner mitochondrial membrane? We call them TIM. Okay. So I hope you got that, TOM and TIM. Okay. But who's the master guru? Okay. You now know TOM, you know TIM, but who's the master guru? Okay. Dapat yan, lalabas yan sa exam. Ha? I swear you're going to hear my voice during the exam. Now, please remember this by heart. Okay. For a protein to pass through the complexes, it has to be in the unfolded state. And this is made possible by binding to chaperone proteins. This now involves heat shock protein 70. So question, what specific chaperone is required for proteins to enter the mitochondrial membrane? HSP what? Needed to enter the mitochondrial membrane. That is specifically HSP 70. You need to memorize that. I hope you got that. I memorized that. That's HSP 70. So here, memorize this by heart. HSP 70. So here, if you are to take an exam, what are the roles of a chaperone inside the mitochondria? So you have imported proteins. We call them imported because the mitochondria cannot synthesize its own protein. It begins with translocation, which is the passage of one protein from the inner to the outer mitochondrial membrane. Sorting occurs, then folding. Okay? So remember, pagpasok sa protein into the mitochondria. When the protein enters the mitochondria, in what state or form is it? Folded or unfolded? Yung pagpasok, the entry, it is in an unfolded state. Then it undergoes folding, then assembly, then lastly, degradation. Okay, I hope you understood this. This is a chapter in the textbook. This is seldom touched in lessons, but the reason I'm giving it as a supplement is because there were questions on diseases. There were questions on heat shock protein, questions on chaperones, question on unfolding, misfolding, okay? So here, some general features of protein import. Okay, yung pag-import ng protein to the organelle. I'm giving you this assignment to go over this table, okay? Just read through it. You don't have to go crazy memorizing it. Now, I want to mention this substance known as cariopharines. There are two types of cariopharines. We have importins and exportins. Okay, importins and exportins. Okay, so here, the importins will interact with molecules and they will dock in what we call the nuclear pore complex. I'll show you a picture of that in a while. Then we have the exportins, which will export macromolecules from the nucleus. Okay, so question, which will interact with the nuclear pore complex? Is it importins or is it exportins? Which will interact with the nuclear pore complex? It is the importins. Okay, now here, this is an example of how proteins will dock, okay, in the membrane. So this is the peroxisomal membrane here. And you can see proteins docking, okay? That's just an example of this process. Now, this is going to come out. I swear this is going to come out. These are the disorders of peroxisomal abnormalities. If you notice, this is chapter 49, way in the back of the book. So first is Zellweger syndrome. What is the problem in Zellweger syndrome? Is it short chain or very long chain fatty acids? What is the problem in Zellweger? In Zellweger, you need to quantify it has to be very long chain fatty acids. Then we have adrenoleukodystrophy. Okay. Then we have Refsum's disease. Okay. So what accumulates in Refsum's disease? What acid accumulates in Refsum's disease? What acid accumulates in Refsum's disease? What it accumulates in Refsum's disease? Okay, very good. It's phytanic acid. Okay, and here's adrenoleukodystrophy. So just take note of these three. These are the classic peroxisomal disorders. Okay, so these are the peroxisomal disorders you should bring with you to the boards. Zellweger, Refsum, adrenoleukodystrophy. In the older edition, they will just mention Refsum. But in the newer editions, they will specify it's infantile Refsum's disease. Okay? So in Zellweger, there's actually a mutation of the PEX genes. Okay? The PEX genes or the peroxines. Now, allow me to go back to this illustration. You see this? PEX, PEX, PEX. That is what is mutated, okay, in Zellweger syndrome. The peroxines, they are the PEX. So the problem with Zellweger is mutations in PEX. So what are the manifestations of Zellweger? So there is usually death within a year. There is profound neurologic impairment, which includes failure to thrive, hypotonia, seizures, okay? And there are abnormal biochemical findings as a result of problems with your peroxisomes. Now, this is the biochemical finding. There's accumulation. You need to remember this. It's very long chain fatty acids. You need to specify it because the other amino acids, short chain, long chain, are not involved. It's only the very long chain. Okay? And there's also going to be abnormalities in the synthesis of bile acids, and there's going to be reduction of phospholipids such as plasmalogens. Neonatal adrenoleukodystrophy. So this presents with white matter disease and problems with the adrenal gland. There's hypotonia, okay, white matter disease, that's the leukodystrophy. There's also problems with hearing. There's also problems with vision. So it's hearing and vision, which is the problem. Okay, hearing and vision. Now, let me show this. Refsum's disease. So it lacks the enzymes that will break down phytanic acid. As a result, there's going to be elevated levels of phytanic acid. Okay, elevated levels of phytanic acid. So please take note of that. Okay, so here, just some photos. Okay, this is Refsum's disease. You have bony abnormalities, you have vision problems here. And this is what we call the referee, which we commonly call ref, or refsum, referee, because the referee will prevent players from fighting. Fighting is phytanic acid. Okay, so it presents with retinitis pigmentosa, problems with smell, particularly anosmia. There's hearing loss, so the special senses are affected in Refsum's disease. There can also be ataxia. There can also be ichthyosis, or what we call the fish skin. Okay, the skin looks very scaly, like the skin of the fish. That's ichthyosis. There are skeletal abnormalities like the pictures I showed you earlier. The toes, they also have cardiac anomalies, particularly cardiac arrhythmia, as well as cardiomyopathy. So please take note of that. Cardiac arrhythmia and cardiomyopathy. Okay? So please take note of this. And some tables, which I just included there. Okay? The only thing I want to highlight here regarding the signal peptides is that it's methionine, which is the amino terminal amino acid. Okay, just remember that it is methionine. Now here, this one came out. We have chaperones, as well as enzymes, which are involved in folding. So I want you to be familiar with calnexin and calreticulin. Okay, calnexin and calreticulin. Calnexin and calreticulin. So please take note of that. Okay, calnexin and calreticulin. So what is calnexin? So cal or calcium, it's a calcium binding chaperone. This is found in the endoplasmic reticulum membrane. And it plays a very important role in binding with the MHC, or what we commonly call the major histocompatibility. Okay, major histocompatibility complex. So aside from binding with the major histocompatibility complex, okay, it also binds with plasma proteins. Okay, it also binds with plasma proteins. Okay, so please take note of that. And of course, we have calreticulin. Okay, this is a membrane bound, it is a non-membrane bound protein. It's also a calcium binding protein. Okay, so there are two, calnexin and calreticulin. Okay, now this table is found in your book. You have here some mutations, okay, of misfolded proteins. So one is alpha-1 antitrypsin deficiency. This is, of course, associated with liver disease. And what respiratory disease? What respiratory disease is associated with alpha-1 antitrypsin deficiency? Okay, very good. It's emphysema. Okay, it is emphysema. Cartaginer syndrome is associated with deficiency in dynein. Okay, so here's Chidiac Higashi. Here's cystic fibrosis. Protein is the cystic fibrosis transmembrane regulatory gene. Gauchers, it's beta-glucosidase. Then we have here the eye cell disease. Okay, here they mentioned phosphotransferase. Okay, and we have Tay-Sachs, which mentioned in the past. This is hexosaminidase. But there's something confusing because in this table, they write here beta-hexosaminidase. Okay, this is supposed to be hexosaminidase A. So we now wind up with the degradation of proteins. So there are two major pathways for protein degradation. One, you have the lysosomal proteases. Second is ubiquitin. What is the major difference? What is the major difference between these two pathways? It is ubiquitin, which utilizes ATP. Ubiquitin is ATP dependent. Okay, so you'll be having a self-directed learning, a self-study assignment to read and watch the glycogen storage diseases. So please take note of that. So here, winding up, let me mention the proteasomes. Okay, the proteasomes is the site where ubiquitinated proteins are degraded. So again, the proteasomes, you're going to find them in the cytosol. They are the site where ubiquitinated proteins will undergo degradation. And as mentioned earlier, what differentiates the ubiquitin from the protease pathway is ubiquitin utilizes ATP. Here are the ATPases. It is ATP dependent. So I ask you, winding up, why is the proteasome important? The proteasome plays an important role, particularly in the degradation of viruses, as well as it plays an important role in the major histocompatibility complex. So our immune system, it plays an important role. That's the role of your proteasome. Now, here's another table, which I want you to be familiar with. Okay, this is the clathrin that came out in the Fisher exam. Okay, this is involved in post-Golgi apparatus transport. So this would be one of your vesicular proteins. Other vesicular proteins is COPY, okay, CAP1 and CAP2. Okay, we call them vesicular proteins. Now here, I just want to show you this clathrin. Ito yung lumabas sa Fisher exam. So clathrin is a vesicular protein which plays a role in carrying cargo to the lysosomes. Okay, so clathrin, as you can see here, okay, has 3 interlocking spirals. And clathrin is a very important protein. Okay, it's a very, very important vesicular protein which plays a role in transport. Okay, then for the COPY vesicles, So what's the difference between the two? COP1 is retrograde transport. When you say retrograde transport, it's from the Golgi apparatus to the endoplasmic reticulum. COP2 is involved in anterograde transport. That means from endoplasmic reticulum to the Golgi apparatus. The normal direction of transport or movement is from Golgi to endoplasmic reticulum. That is anterograde. I know that that is retrograde. Okay. Then we have here endoplasmic reticulum to the Golgi. You can do it bidirectional. Okay. So in this case, just remember COP1 and COP2. So lastly, we have some steps in the life cycle of vesicles. We call them budding, tethering, docking, and membrane fusion. Okay. The one that usually pops out in the boards is docking. Okay. Can you give me examples of docking proteins which are associated with acetylcholine? Docking proteins. We have what we call the snare, snap. Okay. We have synaptobrevin. We have synaptophycine. These are the snare proteins. These are usually affected by what toxin? What toxin usually affects the docking proteins, the snare, the snap? Okay, very good. It's botulinum toxin. Okay, botulinum toxin. So here, these are just the processes. Okay, so there's the budding, there's the docking, and in here, lastly, there's the fusion. I just want you to be familiar with that. So here, this is the snare proteins. We have the SNAP receptor. Okay, the SNAP receptor plays a very important role in fusion. Okay, because fusion has to occur for final transport to happen. So here, I just want to end with this. Okay. Here's the copy. Here's the snap. And here's the snare. Okay, you don't have to memorize this. I just want you to know that they exist. Okay. Now, let's go to the back of the book. This is now where the exam comes in. So in the synaptic vesicle, there's a snare protein known as synaptobrevin. This binds with botulinum toxin. And once botulinum toxin binds with synaptobrevin, this will inhibit the release of acetylcholine. So there will be muscle flaccidity. There will be paralysis. This is what you need to memorize. Okay? You need to memorize synaptobrevin. So lastly, there is a drug mentioned in Harper's called geldanamycin. Okay? This drug activates, it's an antibiotic. It activates heat shock proteins. Therefore, it will promote proper folding of proteins. Geldanamycin. I've never met this in my life, but it is in bold in your Harper's. Okay, so your personal study is going to go over the glycogen storage diseases. Okay, and I want you to review this session. Study your handouts. You don't have to open the book anymore. This is already summarized. Are there any questions? May tanong ba? Let's see everyone.